Hello everyone, good morning all of you. This is Dr. Sudarshan Sambhajirao Pedge, Assistant Professor, Department of Zoology, Kallasasya Rasika Mahavidyalaya Devni, your zoology companion, and welcome to my lecture. In the last lecture, we also completed the formation of spermatid. The first stage of spermatogenesis is the formation of spermatid. Now today we will start the another next point of this topic, that is the spermiogenesis. Okay, so as well as the second stage of the spermatogenesis is the spermiogenesis. Okay, so let's get started today's lecture. In the spermiogenesis, the transformation of the spermatid into the spermatozoon is called the spermiogenesis. Okay, what is meant by the spermiogenesis? Spermiogenesis, it means the transformation of the spermatid into the spermatozoon is called spermiogenesis. Okay, so the spermatid is in the form of a typical cell containing the nucleus, Golgi bodies, mitochondria, centrioles, etc. Okay, it means the one particular cell organelles present in the spermatid. Okay, in the spermatid, what type of cell organelles is present? The nucleus, Golgi bodies, mitochondria, and centriole. Okay, so but it contains only haploid number of chromosomes. Okay, so uh, we have seen in the last lecture the spermatid contain only one haploid number of chromosomes. Okay, so the during the differentiation of the sperm, all these organelles undergoes changes. Some changes we have seen in the spermiogenesis, or the some changes we have seen in the spermatids. Okay, so first changes, the nucleus is gradually diminishes in its size by losing water. Okay, it means the nucleus gradually so decreases in size. Okay, so why? Because the losing of water. Okay, so another the second one point, important point is the chromosome become a concentrated and are closely packed. Okay, you have seen in the cell of the spermatid, the cell is the one particular concentrated or closely packed with the chromosome. Okay, so that is the one particular. Another one more point is here, that is the in the during differentiation of the sperm. Okay, so another one third point is the all the achromatic substances. Okay, that is the nucleolus and RNA is disappear. Okay, you know very well the some achromatic substances we have seen in the all types of cells. Okay, so another this cell, that sperm cells we have seen in the nucleolus and RNA. Okay, that nucleolus and RNA in the spermiogenesis, these nucleolus and RNA is totally disappear. Okay, so this is the one particular third undergoes changes in the differentiation of the sperm. Okay, another one more point here, the fourth point, the nucleus becomes an elongated. Okay, so the nucleus, if you seen in the spermiogenesis process, that time the nucleus is elongated. Okay, so another next is the Golgi bodies. Okay, you have seen in the Golgi bodies, the another you have seen next slide. So you have seen here in the Golgi body develop into a acrosome. Okay, in the acrosome formation, Golgi bodies. Okay, in an early spermatid, the Golgi bodies are in the form of a membranous vesicle. Okay, so and it arranged around a group of vacuoles. You have seen here the arranged in the group of vacuoles. Okay, so the small vacuoles gradually fuse together to form a large vacuoles. Okay, you have seen here the small vacuoles is fused together and formation of large vacuoles. Okay, so it is now called as a acroblast. Okay, so we have seen that fuses of Golgi bodies. We have seen we have call it as that is acroblast. Okay, and in the inside the acroblast, a dense body called the proacrosomal granule. Okay, that is the proacrosomal granule. Okay, you have seen here the proacrosomal granule. Okay, why? Because the dense body form. Okay, and that dense body inside the acroblast, a dense body is called the proacrosomal granule. Okay, is developed. So the whole of the acroblast spreads over the front part of the nucleus. Okay, you have seen here the front parts of the nucleus. Okay, so so the proacrosomal granules enlarges to form the acrosomal granule. Okay, so in the acroblast is now called the acrosome. Okay, so we which forms the cap of the sperm. Okay, so the acrosome and the nucleus together constitute the head, and the remnants of the Golgi bodies degenerated and eventually get discarded. Okay, so this is the one particular uh, process of the Golgi bodies. Okay, it means the Golgi bodies develops in the um, spermiogenesis. Okay, another one more point we have seen here, the centrosomes. Okay, so the centrosomes of the spermatid contains the two centrioles. Okay, so you have seen in the next slide. Okay, so you have seen here the centrosomes uh, centrosomes of the spermatid contains the two centrioles. Okay, and they move towards the nucleus and occupy a position behind it. Okay, so a depression is formed in the posterior surface of the nucleus, and one of the two centrioles becomes placed in this depression. Oh, okay, so this is the proximal centriole. Okay, we have seen here the proximal granule, sorry, proximal centriole. You have seen in the after the proacrosomal granule, you have seen here the one with some dot like structure, that is the proximal centriole. Okay, so the proximal centriole, the other centriole is called the distal centriole. Okay, you have seen the distal centriole is there. Okay, uh, and that distal centrioles takes up a position behind a proximal centriole. Okay, after the proximal centriole, you have seen in the distal centriole. Okay, so another one more point here we have seen here, the distal centriole develops a filament like structure, and that filament like structure is called the axial filament. Okay, you have seen after the distal centriole one filament like structure is there. Okay, so that is the axial filament. Okay, so it gradually elongated and forms the tail by developing a cytoplasmic sheath. Okay, so you have seen the one particular tail like structure it will be formed. Okay, that is the axial filament. Okay, the distal end of the axial filament is free from the cytoplasmic sheath, and it is named is the end piece. Okay, so another one more point we have seen here, the that is the mitochondria and manchit. Okay, so you have seen here the mitochondria and manchit. The mitochondria of the spermatid are aggregated together to form a large mass in the region of the centrioles. Okay, so this is called the mitochondrial cloud, and in mammals the mitochondria cloud becomes a twisted spirally around the proximal part of the axial filament. Okay, so this is the one particular. However, you have seen in the other animals the spiral arrangement of the mitochondria is not found. Okay, so and they may be arranged in a two bands on either side of the proximal parts of the axial filament, or clumped together in a compact mass. Okay, it is the one particular the region forms of the middle piece. Okay, so after the middle piece, the around the surface of the middle piece, the cytoplasm forms a condensed sheath, and then condensed sheath is called the manchit. Okay, so that is the one particular process. The cytoplasm flows backwards and forms a thin layer around the nucleus, middle piece and a tail. Okay, so that is the one particular total process is called the spermiogenesis. Okay, so this is it for today. Thank you very much.